Good day, dear students. Today we'll tell about very important and very interesting part of amino acid metabolism, about their conversion to a specialized product. Just see that all of the amino acids that can appear in our body, they all can be acquired through the breakdown of our own intracellular proteins, or can be ingested dietary proteins. And these amino acids can enter three metabolic routes within our body. First of all, they can be used for biosynthesis of tissue proteins, our own proteins, or can be created to amino acid derivatives, and we will tell about this pathway today, or they can be catabolized into their functional group and carbon skeletal, and this process involves reactions of deamination and transdeamination or irreversible deamination reactions with the releasing of ammonium. And this ammonium, after that, this ammonium enters the urea cycle and is converted into urea and excreted through the urine. Each carbon skeleton of deaminated amino acid follow the unique metabolic pathway and can be broken down into the different intermediates and pyruvate and acetyl-CoA and oxaloacetate and so on. And these compounds can be all completely oxidized to carbon dioxide and water with energy production through the citric acid cycle. Let's to see it better on the next our slide. You see that all of our 20 amino acids, they can be all converted into acetyl coenzyme A, or can be converted into pyruvate, oxaloacetate, fumarate, succinyl coenzyme A, alpha ketoglutarate, you see, and they can be all finally degraded to carbon dioxide and water with energy production, or can be used for biosynthesis of glucose or ketone bodies. In the spite of this common fate, amino acids are classified as glucogenic and ketogenic according to the type of their intermediate metabolites. The ketogenic amino acid gives rise to acetoacetate or acetyl coenzyme A, from which acetoacetate can be synthesized. The glucogenic amino acid, which give rise to pyruvate or oxaloacetate, fumarate, succinyl coenzyme, alpha ketoglutarate, you see, intermediates of the citric acid cycle, and all of them, they can serve as a substrate for gluconeogenesis, with final result glucose synthesis. Really, only two amino acids, you see, we mark them, these two amino acids, leucine and lysine, they are clear, purely ketogenic. These aromatic amino acid, together with isoleucine, they are both, you see, in tryptophan, phenylalanine, tyrosine, isoleucine, they are both glucogenic and ketogenic. It means that during their catabolism, their carbon skeleton can be cleaved into two parts, and one part is used for ketone body synthesis, another part for glucose synthesis. That's why they are mixed glucoketogenic. All other amino acids, they are glucogenic. Let's again repeat them. You see, two of our amino acids, leucine and lysine, they are ketogenic. 5 glucoketogenic. And 13 last amino acid, they are glucogenic amino acids. But you see, you already know that all our amino acids can be used or for biosynthesis of our own protein de novo, or can use for fat or metabolism or glucose synthesis. But together with this amino acid can be converted to different very important substances, amino acid derivatives. And today we will tell about this way. This is our topic for this lecture. See here that of course, all of these amino acids, all 20, we can use for protein synthesis. They can be or glucogenic, or ketogenic, or both. But see again on this picture how many different, very important substances it is possible produce from these amino acids. And purines and pyrimidines, that's why nucleic acid, and pigments, and different biogenic amines, between them many hormones and neuromediators, and heme, and choline, and phospholipids, and creatine, and carnitine, and many, many other. And I will tell you about this pathway, this using of unusual, very important using of amino acids in our body. We start to discuss this conversion of amino acids from two sulfur-containing amino acids. We have only two of sulfur-containing amino acids, cysteine and methionine. These amino acids are metabolically closely related. And let's first of all tell about the metabolism of cysteine. First of all, you see, of course, cysteine, it's a protein's amino acid, but cysteines play a special role in the proteins. If you remember this famous disulfide bond, which is so important for the correct formation and stabilization of three-dimensional structure, only cysteines took part in the formation of this strong covalent disulfide bond. That's why cysteine plays a very important role in the normal proteins folding. And two molecules of cysteines, you see this is cysteine and this, they interact one with another, form this disulfide bond, and the name is these two cysteine residues to form a cysteine molecules. Except of these, many proteins and enzymes, they have in their active site cysteine, and it's this SH group involves in the catalysis in many reactions. Cysteine participate in the synthesis of glutathione. It is a typical tripeptide containing gamma-glutamic acid, cysteine, and glycine amino acid. So it consists from 3 amino acids. That's why I will show you the biosynthesis of glutathione and its function a little later when we will tell about glycine metabolism. Cysteine also serves as a precursor of the thiaethamine fragment of coenzyme A. See, this is a structure of coenzyme A, which consists the base of this coenzyme A, of course, pantothenic acid, vitamin B3, but pay attention on this part, you see this, it is a derivative of cysteine, result of cysteine decarboxylation, beta-mercaptoethylamine. See, after decarboxylation of cysteine, we produce this group, and this cysteine's SH group, it's a place for acyl or acetyl group attachment. Cysteine is practically the only source of the urine sulfate. And another important way of the using of cysteine is a synthesis of taurine in animal tissue and in our organism the same. Synthesis of taurine, I will show you full synthesis of taurine, you see, occurs by decarboxylation of cysteine derivatives or cysteic acid or cysteine sulfonate. So here may be two waves of taurine production. First reaction is oxidation, and from cysteine we produce cysteine sulfonate, after decarboxylation of which hypotaurine is produced, and next oxidation of hypotaurine lead to taurine synthesis. Or maybe another way, we continue oxidized cysteine sulfonate and produce cysteic acid, and now already decarboxylation of which lead to taurine synthesis. You have to know this way or this, but we will ask you during our classes the reactions of taurine synthesis. And this molecule is important for us because taurine has many different functions. It is essential for the synthesis of the bile acids in the liver. In addition of this, it is very important in the cell as an antioxidant agent. It is used to reduce lipid peroxidation, and it binds to hypochlorite anion with the formation of chloramine complex. And it is play a special role for the normal brain development. You can read here all functions of taurine. If we tell about metabolism of methionine, it's, you see, rather complex. First of all, methionine, the same plays a special role in the proteins. Why? What a special role? I think that you remember it. Methionine is caused by a start codon, AUG. Thus, methionine is involved in the initiation of the translation processes. All our proteins, they start from methionine synthesis. It is the first amino acid in them. After that, they can be cleavage. And we already discussed that, we not tell, but now I won't tell you that methionine is essential amino acid, but cysteine, it is relatively essential. Why? Because cysteine can be produced from two amino acids, from serine, which gives the carbon skeleton, and the donor of sulfate, this sulfur atom for cysteine is methionine. It's one of the functions of methionine. It's supporting biosynthesis of cysteine. Methionine is glucogenic amino acids, and one more very important function of methionine, it is a donor of this methyl group. This methyl group of methionine is a mobile one, you see, carbon fragment, and it is used for the synthesis of a number compounds. You see how many, because we can take this methyl group from methionine and gives them to these compounds. And name of this process is transmethylation reactions. This reaction has very important metabolic significance. But the methyl group in clear, this simple methionine molecule, is tightly bound to the sulfur atom. That's why the direct donor of this one carbon fragment is not methionine itself, but its active form. And first of all, necessary to activate methionine, and the name of its active form is S-adenosine methionine, SAM. Let's see what happened during this activation, reaction of methionine activation. See here. And a little after that, we will tell, after discussion of this reaction, we will tell about a few examples of this transmethylation reaction, due to which an carnitine and epinephrine can be produced. You know that we use methylation for biosynthesis of nitrogen bases. We will tell about detail about this pathway of creatine synthesis. Choline, it's known for your molecule because choline we use, and for choline we use and for acetylcholine synthesis and phosphatidylcholine synthesis. S-adenosine methionine took part in production of melatonin, different glucosaminoglycans, and it's the same took part in the different reactions of detoxification, because took part in methylation reactions in the liver. Now let's tell about the reactions of methionine activation. The active form of methionine is, as I tell already, S-adenosine methionine. And the sulfonium form of the amino acid were formed by the addition of methionine to ATP. During this reaction, it's an absolutely unique reaction, because during this reaction from ATP we remove all 3 phosphate group. We don't know another example of this variant. And now we will transfer our methionine to adenosine only. You know that adenosine, this is a sum of adenine plus ribose. This is adenosine. And we produce, you see, adenosine methionine. And our adenosine is attached to the sulfur. That's why name of this is S-adenosine methionine. SAM. This already active form of methionine took part in transmethylation reaction. Now we very easy, later I will show you again, can take this sulfur group and donate this sulfur group to any substrate and produce, you see, this is happened, the methylation of substrate, but together with this, our methionine is converted to after this reaction into S-adenosine homocysteine. But about this later. Now see on this active form of methionine. This reaction, formation of S-adenosine methionine, is catalyzed by adenosyl transferase, this enzyme. And this enzyme, adenosyl transferase, present in all type of our cell. In the S-adenosine methionine, for us interesting this group, you see, is sulfur now, has a positive charge, and it's a trivalent sulfur, you see. And due to this methyl group now is unstable group and has very high activity. Very easy now we can transfer this group from donor to different substrate acceptors. That's why name of this is active methionine. Now, during the cleavage of this, as I tell already, S-adenosine methionine is transferred to S-adenosine. I tell it that very easy to take this methyl group to homocysteine. But first of all, I want to show you one of example of transmethylation reaction. During the lecture, we will tell about almost all of them, because later we will tell and about epinephrine synthesis and about choline synthesis. But here you see the same very important for us pathway, biosynthesis of creatinine. We will see this picture not one time because 3 amino acids took part in creatine synthesis. You see, first amino acid is arginine, second glycine, and during this step, you see, S-adenosine methionine took part in methylation. But let's tell about this pathway step by step. First reaction occurs in the kidney. Pay attention that it's a not very long way, but very interesting because it takes place in different organs. Okay, let's continue. So first reaction, it's a transfer of a part of arginine from first amino acid arginine to glycine. During and arginine glycine transamidinase catalyze this reaction. This enzyme present in the kidneys. We take this amidine group of arginine, see here, and attach this amidine group to nitrogen of our glycine. This part of arginine has name ornithine, and about the function of ornithine you will listen next lecture. It is a part of the urea cycle, but in this pathway the name of the product is guanidinoacetate. Guanidinoacetate, after that, with the bloodstream, is transported to the liver. And in the liver already, we used, we have the continue, the next reaction. First of all, happen the phosphorylation of, first of all, we form creatine in the liver, and during the creatine happens the methylation. You see, we take one methyl group from our S-adenosyl methionine and attach this methyl group to this nitrogen, and here pair CH3 group, and name of this product is creatine. This enzyme, the enzyme which catalyzes this reaction is transmethylase. After that, creatine is transported in the bloodstream to the muscles or brain cells, where we have another enzyme, creatine kinase. And we will tell about this enzyme especially a little later. But under the action of this enzyme, very important molecule is produced, creatine phosphate. This molecule, creatine phosphate, is a form of high energy compounds. It's a rich of energy. Pay attention. You see, this is creatine phosphate. And enzyme which catalyze this reaction, creatine kinase, this enzyme is localized in the cytosol and in mitochondria of the cells, and this enzyme has organ specificity, and we you know this enzyme. Normally activity of this enzyme in the blood is very known, and you know that found three isoenzymes forms of creatine kinase: MB, and BB. form is characterized for muscles, skeletal muscles. MB form for the heart, and BB form for brain cells. Creatine phosphate plays an important role in providing energy to working muscles during the initial period. And as a result of these non-enzymatic reactions, dephosphorylation, mainly this reaction occurs in the muscles, of course, creatine phosphate is converted to creatinine. And creatinine after that is excreted in the urine. And the daily release of creatinine in each individual is a constant, and it's proportional to the total muscle mass. And determination of the content of creatine and creatinine in the blood and urine is used to characterize the intensity of muscle work. And especially it's important for the sport medicine and in some pathological condition. Determination of the activity of enzyme creatine kinase, and of course it's first of all its isoenzymes form in the blood, is used in medicine for diagnosis of different diseases such as myocardial infarctions or myopathias or muscular dystrophies and other. Pay attention that this way start in the kidney, continues in the liver, and final reactions we can see or in the muscles mainly or in the brain. Brain the same can use creatine phosphate like additional source of energy. And I will show you this picture. And when we tell about glycine and when we tell about the role of arginine in our body. Now, you see that there are very many reactions of methylations in our body. And this reaction is important for us. And they are very intense. This causes a large consumption of methionine since it is an essential amino acid. And you see that methionine cannot be synthesized in our cell. And we take methionine only from food proteins. And in this case, it is regard the possibility of methionine regeneration with the participation of any non-essential amino acids, serine, for example, or glycine, and this regeneration is a great important for us. And as a result, you know that as a result of the cleavage of methyl group as adenosyl methionine, you see, is as adenosyl methionine. You see, this is activation reaction. Again, let's repeat again. Activation of methionine and formation of its active form as adenosine methionine. And when it took part in methylation reaction, you see, give this methyl group to substrate. S-adenosyl methionine is converted to S-adenosyl homocysteine. After that, this S-adenosyl homocysteine can be cleaved by hydrolase to adenosine, into adenosine and homocysteine. And now you see possible the remethylation of homocysteine again into methionine. For this reaction, we used enzyme homocysteine methyltransferase. We add methyl group. This methyl group, this one carbon fragment, we can take or from serine or from glycine. They are not essential. We can produce it easily, and the amount in our body is enough for reactivation of essential methionine. And you see that the donor, it's we first of all take this one carbon fragment from another amino acid, give this one carbon fragment from tetrahydrofolate. It is a derivative of vitamin B9, folic acid, active form of folic acid, and after that use this methyl group for resynthesis of methionine. And pay attention that intermediate carrier of the methyl group in this reaction is a derivative of vitamin B12, methylcobalamin, which acts as a second coenzyme of this reaction. And later, when we will tell about the vitamins, you will see that both of this vitamin and B9 and B12, they took part in the metabolism of one carbon fragment. And here you can see one of their using. Later, I will every time ask you what amino acid took part in choline synthesis or epinephrine synthesis, and I hope that now you already can recognize it and to answer me that, of course, active methionine, SAM, S-adenosine methionine. Next amino acid that we will discuss with you today, they are the simplest, your lovely amino acid, glycine and serine. These amino acids, they the same perform various and very important functions in the human body. They play important role in the synthesis of many biological importance compounds. Like nucleotides, coenzymes, heme, complex lipids, creatine, and other. Both of these amino acids, they are non-essential, they are glucogenic, that's why both of them, they are used and for protein synthesis and for glucose. Well, let's start. And as usually, we start from glycine. You see, glycine. Glycine, I already tell, it is a non-essential amino acid. Glycine can take part in biosynthesis of other amino acids, serine and threonine, and the reaction of the synthesis of serine from glycine, it is reversible reaction. See here, that's why they can interconvert it one to another. And if, for example, serine is converted to glycine, serine in this case is a donor one carbon fragment. If we want to produce serine from glycine, necessary to add one carbon to glycine. So, and again, I want to show you that this reaction is reversible. That's why we tell about interconversion of serine and glycine one to another. Glycine plays a special role in some proteins. For example, in collagen, concentration of glycine is very high. Its third amino acid in the collagen is glycine. Glycine is necessary for the heme synthesis. You already know I showed you that glycine took part in the synthesis of creatine. It took part in the first reaction in creatine synthesis together with arginine. I will show you that today that glycine is necessary for purine synthesis. Glycine took part in biosynthesis of bile acid. Already today we'll tell, and now I will show you reactions of glutathione synthesis, where three amino acids and on one took part, and one of them is glycine, and we will tell about the function of them. Glycine we can use for conjugation with different xenobiotics, and it's a way of the detoxification of them in our body. Different xenobiotics and drugs the same. And glycine itself is the most important, after GABA, of course, inhibitory neurotransmitter in the spinal cord, diencephalon, and some part of the brain. And high level of glycine in the blood, plasma, and urine usually indicate impaired brain function. You see how many functions of this glycine. Now, let's see. I promised to show you glutathione synthesis, but first of all, I again want to repeat you the function of glycine in creatine phosphate synthesis. We discussed this way. I only repeat it and show position of glycine in this pathway together with methionine. And now you see synthesis of glutathione. Glutathione is unusual, atypical tripeptide. Why atypical? Because it's a tripeptide. It consists from three amino acids, you see, glutamate, cysteine, and glycine. Only pay attention on the first reaction, formation of the first bond in this tripeptide. You know that classical peptide bond is formed between alpha carboxylic group of first amino acid and alpha amino group of the second amino acid, but not in this case. Here we form bond between gamma carboxylic group of our glutamate and alpha carboxylic group of cysteine. And form, you see, this unusual dipeptide. Pay attention on this bond. It's not classical bond between this group and this. It's a bond is formed between gamma carboxylic group and amino group. Next amino acid, our glycine, we attached traditionally without problem. Here already we will, you can see reaction between alpha carboxylic group of cysteine and alpha amino group of our glycine, and we form triapeptide with a special name glutathione. And you see that you can use this pathway for the explaining and cysteine metabolism and glycine metabolism and glutamic acid metabolism. What is the function of glutathione in our body? Very many functions. First of all, it serves as an antioxidant. We have a glutathione and depend enzymes. It took part in conjugation with different xenobiotics, with conjugation with the drugs, which lead to increases of their solubility, and such way we can tell about detoxification of them. You know that biochemically detoxification means increasing of solubility. Glutathione is involved in amino acid transport across cell membrane. And glutathione can serve as a cofactor for some enzymatic reaction. It is a cofactor of a group of enzymes. I promised to show you the role of glycine in purine synthesis. And really, you see, in purine synthesis took part many amino acids. For example, this nitrogen we take from aspartate. These two nitrogen we take from glutamine. This carbon from carbon dioxide. These two carbons, it's maybe or from methionine or from glycine or from serine. They are one carbon fragment, which carries, each is incorporated to purine from tetrahydrofolate. And pay attention on this pink color group. This is a full glycine. You see that all atoms of glycine, they are incorporated into purine molecules. That's why glycine plays very important role in RNA and DNA synthesis. But as we discussed already, almost always different metabolic pathway can have any disorders, problems with the enzymes, deficiency of enzymes, or abnormal activity of them. The same we can tell about some hereditary disorders of glycine metabolism. Currently, several diseases which are associated with disorders of glycine metabolism, and they are based or on the lack of any enzyme or a defect in the transport system of this amino acid. But in all cases, we can see accumulation of glycine in the body, and first of all in the blood. And name of the situation is a hyperglycemia. It is increased concentration of glycine in the blood. Usually we can see due to the defect of in glycine cleaving enzyme system. And the most severe manifestation of hyperglycemia is a severe brain damage, convulsion, hypertension, and respiratory failure. And it's a pity, but it is often fatal. A pair of glycine in the urine, glycinuria, is characterized by the increased excretion of glycine in the urine. You see? And one of symptoms of this disease is the formation of the oxalate, kidney stone. See the picture. Excess oxalate is of endogenous origin. It is obtained, you see, from glycine, upon deamination of which, first of all, we form, you see, this is glyoxylate due to deamination with the removal of this. And after reduction of this glyoxylate to oxalate. The cause of this glycinuria is obviously a violation of the glycine reabsorption in the kidney. In the future, it may be lead to bilateral formation of the oxalate stone in the urinary tract, and it can progress and finally lead to different nephrocalcinosis, and urinary tract infection can develop, and the patient even can die in childhood because it's inherent problem, and usually or from renal failure or from hypertension. So it's a dangerous situation. Now serine. Serine is also non-essential glucogenic amino acid. You see, glucogenic amino acid. Serine, the same, of course, used for protein synthesis, and serine, the same, plays important role in the proteins. We have a number of serine proteases, where serine is a part of the active site of this enzyme, because serine has very active OH group. We took part in catalysis of different reaction. We already tell about converted of serine into glycine. Moreover, you see that serine can take part in formation of phosphatidylserine. Combination of serine with phosphatidic acid gives us these important phospholipids, which play a special role in the formation of our membrane. During our classes, we wrote already reaction of serine decarboxylation, which lead to formation of ethanolamide, and you see how many very important substances we can produce from ethanolamide: choline, and from choline corresponding sphingomyelin, phosphatidylcholine, acetylcholine. Ethanolamide, the same, lead to formation of phosphatidylethanolamide. And we will write now with you some of these reactions. Serine is, I already tell, is non-essential amino acid. Serine can be synthesized an intermediate product of glycolysis, 3-phosphoglycerate, and the amino group after that to this 3-phosphoglycerate we take from glutamic acid. What else interesting you have to know about serine? Serine does not take part in transamination. It has a special way of deamination by dehydration, directly deamination. And as we tell already, one of the function of serine, it can be a donor of one carbon fragment. This its group, CH2OH group, is readily transferred to tetrahydrofolate. And tetrahydrofolate, I already tell you, is a coenzyme of one carbon group transferases. And due to this, as this donor, serine can take part in many different reactions. And due to this, when it becomes a donor of this group, serine is converted to glycine. And we already tell that glycine itself can be a donor of one carbon group. And all of these reactions, they are reversible. Let's see any of these reactions. For example, you see that synthesis of acetylcholine. Serine After decarboxylation of serine, with serine decarboxylase and vitamin B6 pyridoxal phosphate took part in decarboxylation, we produce ethanolamine. Now see that methylation of this group, this nitrogen in ethanolamine, lead to production of choline. Let's count how many methyl group here. 1, 2, 3. All of this methyl group we take from, yes, of course, from methionine, S-adenosine methionine. But one methionine gives only one methyl group. That's why for choline synthesis, we use 3 methionine, active methionine molecules, 3 S-adenosine methionine. All of them, they are converted to 3 homocysteines. Now, choline, we can use for phosphatidylcholine synthesis, but now on the slide we tell about acetylcholine synthesis. You see that after that choline binds with, I hope that you recognize this molecule. I don't write, but now all of you have to know what is it. This is acetyl coenzyme A. And condensation of this molecule lead to production of acetylcholine. You know that acetylcholine is synthesized in the nervous system, nervous tissue, and it serves as one of the most important excitatory neurotransmitters of the autonomic nervous system. Combination of O itself, serine, lead to formation of phosphatidylserine, or if we combine here derivatives of serine, ethanolamine or choline, it leads to production of corresponding phosphatidylcholine or phosphatidylethanolamine. You see, this is a way of their synthesis. Next amino acid that we will discuss today with you, histidine and tryptophan. Maybe their structure is rather complex, but their metabolism is rather simple. But their role in our body continue to be very important. The amino acid histidine is exposed to different enzymes in different tissue, and it is involved in 2 different metabolic pathways. One pathway is simply catabolism of histidine to the end product and with energy production or glucose synthesis, and second is a synthesis of histamine. It is a simply decarboxylation of our histidine. Due to decarboxylation of histidine in the mast cell of connective tissue, histamine is produced. Histamine performs the many different functions in the human body. It stimulates the secretion of the gastric juices, saliva, it increases capillary permeability, causes edema, lowers blood pressure, but only in bloodstream, but it increases intracranial pressure, causes headache. Histidine reduces the smooth muscles in the lung, causes suffocation. It participates in the formation of inflammatory reaction, causes vasodilatation, skin redness, tissue swelling. Causes is allergic reactions, act as a neurotransmitter, and you know that histamine is a mediator of the pain. And very widely we use in the medicine antihistamic substances. They are drugs which blocking the action of histamine, excess of histamine action. No, this reaction is the simplest and usually you have no problem with them. Of course, if you know the structure of histidine. It's simply decarboxylation, and I want to repeat it again. Coenzyme of this aromatic decarboxylase is vitamin B6, pyridoxal phosphate, the name of the active vitamin B6. Tryptophan. Tryptophan is essential amino acid. Tryptophan is both glucogenic and ketogenic. And it is a precursor for the synthesis of important compounds like serotonin, melatonin, and coenzymes of niacin, NAD and NADP. The metabolism of tryptophan is divided into two pathways, kynurenine pathway and serotonin pathway. Here you can see serotonin pathway of tryptophan metabolism. Serotonin, which is produced during this pathway, is a neuromediator, and serotonin is formed in the adrenal gland and in the central nervous system, and normally about 1% of tryptophan is used for serotonin synthesis, converted to serotonin. During the first reaction happened the hydroxylation of tryptophan to its 5 position, you see. And we produce, here I write 2 reactions in one step by explaining you step by step. That first of all happened the hydroxylation of tryptophan, and we produce 5-hydroxytryptophan by hydroxylase enzyme, and then this 5-hydroxytryptophan is converted to serotonin as a result of the action of aromatic amino acid decarboxylase, and we produce serotonin. So here have two reactions, hydroxylation and after that decarboxylation, and we produce, you see, this is serotonin. Serotonin is a biologically active substance with a broad spectrum of action, many different action. It stimulates the contraction of the smooth muscles, has a vasoconstrictor effect, regulates blood pressure, body temperature, respiration. It has antidepressant effect, and according to some reports, modern reports, it can act as a neurotransmitter the same, and it can part in allergic reaction because it is synthesized in small quantity in the mast cell. Serotonin after that can be converted into hormone melatonin, you see, which regulates daily and season changes in the body metabolism, and it is involved in the regulation of some reproductive functions. For example, melatonin inhibits effect, no, it has inhibitory effect on ovarian function. And melatonin also performs a neurotransmitter function the same. If we tell about second pathway of tryptophan metabolism, kynurenine pathway, this pathway mostly occurs in the liver and leading to oxidation of tryptophan and synthesis of NAD or NADP. Here I show you NAD, but you know that if we attach phosphate group to this position, we produce NADP+. We can provide nearly all our nicotinamide requirement from tryptophan. Of course, if we have enough amount of tryptophan in the diet. Normally, about 2/3 of our tryptophan is used for production of these nicotinamide derivatives. Hereditary disorder of this pathway of tryptophan metabolism is named Hartnupp's disease. This disease is characterized by a low blood level of tryptophan and other neutral amino acids, and their elevated urinary excretion. Symptoms, their pellagra-like symptoms are common because, first of all, it depends from the impairment in the synthesis of NAD+, and serotonin from the tryptophan. And if we tell about symptoms, these clinical symptoms includes dermatitis, diarrhea, ataxia, mental retardation. It's the main symptoms. And it can be even the reason of the death, because this reduced equivalence play very important role in our body. Now let's tell about the group of branched amino acid: valine, leucine, and isoleucine. You see, they have branched radical. That's why they are branched chain amino acids. All of them, they are essential, and these three amino acids undergo a common pathway, but they have different final product. I want to show you the pathway of them on the example of leucine. First reaction that happens with leucine is a transamination, and this is reversible reaction, and due to these reactions, these amino acids, they are converted to corresponding keto acid. If we tell about next way, you see, if here you produce, this is keto acid, you see, during the transamination, amino group is changing to keto group, and we produce corresponding keto acid. And next reaction that occurs with this branch chain amino acid is decarboxylation. We remove carbon dioxide, oxidative decarboxylation, and produce branched acyl coenzyme A. If we tell about leucine, leucine is converted to isovaleric coenzyme A. All other, you see, other two branch chain amino acids, they have the same pathway. And that's why you see this scheme, it means a complex for you, but I only repeat this scheme. You see, transamination and oxidative decarboxylation. Here the same, you see, happened. Transamination with them, and we change amino group to keto group, you see. Type of reaction is the same, only different amino acid and different keto acid. And after that happened, the oxidative decarboxylation of them, we remove carbon, you see, carboxylic group like carbon dioxide, and the type of this reaction is absolutely the same like we convert pyruvate to acetyl coenzyme A, or in the Krebs cycle, we the same can see formation of alpha ketoglutarate from isocitrate. This is a type of reaction. And here the same, we have a complex enzyme which consists from multi-enzymes complex. It consists from 3 enzymes and 5 coenzymes. And type of reaction is absolutely the same. Only we produce different acyl-CoA derivatives. Such way, valine is converted, as we tell already, you can see here, valine is converted to, you see, to propionyl coenzyme A. It is a precursor of glucose. If we tell about leucine, leucine is converted to valeric acid, and after that, leucine can be converted to acetyl coenzyme A and acetoacetate. And they are substrate, they are ketone bodies, and the acetyl coenzyme A, they are substrate for fatty acid synthesis. If we tell about isoleucine, isoleucine is degraded to propionyl coenzyme A and acetyl coenzyme A, such as valine is a clear glucogenic amino acid, leucine is a only ketogenic amino acid, but isoleucine, which gives two products, acetyl coenzyme A and propionyl coenzyme A, it is a both. Isoleucine is both glucogenic and ketogenic amino acid. But you see that I especially mark that deficiency, abnormal activity of this enzyme, problem with this reaction, oxidative decarboxylation of derivatives of our this amino acid, lead to a special disease, maple syrup urine disease. And really, the urine is of the affected individuals smells like a maple syrup, burnt sugar. From here the name. But this disease is also known as a branch chain ketonuria or leucinosis. So different names maybe you can read in your book. Maple syrup urine disease, you see, I again want to repeat it. It is due to this defect of this enzyme of the brain chain alpha ketoacid dehydrogenase. And this causes a blockage in the conversion of valine, isoleucine, and leucine to corresponding final product. And as a result, it leads to accumulation in the plasma and appearing of urine high amount of brain chain amino acids and their keto acids. You see, corresponding keto acid. And accumulation of this brain chain amino acid and keto acid it lead to many problems. To impairment in transport and functions of other amino acid, to block of protein biosynthesis. This brain chain amino acid competitively inhibits glutamate dehydrogenase. That's why it leads to disorder of deamination of all our amino acids. Finally, it leads to acidosis, lethargy, convulsion, mental retardation, coma, and finally death within the 1 year after birth, because this is inherited disease. And of course, we want to treat these babies. Is it possible? Yes, but only if we tell about the earliest diagnostics. And you see that this is earlier diagnosis by enzyme analysis within the first week of life is very important in this case. And for this is the estimation of the urinary brain chain amino acid and keto acid, of course, will help us to put correct diagnosis. And if we put this diagnosis, we can treat. Treatment is the simplest. It's a diet with low concentration of this branched amino acid with low concentration, not absent, because as we tell already, these amino acids, they are essential. That's why we must take them from the food for biosynthesis of our own proteins, for the normal development of the organism, but not more than necessary for protein synthesis, necessary to prevent accumulation of this amino acid in the body. And of course, the plasma level of branch chain amino acid should be constantly monitored for adjusting of the dietary intake. Okay. I saw many tell about this. Now you can read about this maple syrup disease or leucinosis, deficiency of this enzyme, brain chain oxidative dehydrogenase enzyme. And here you can see all about what I tell you. You see that if the diagnosis we put very late, and if we don't treat them, it lead to irreversible mental retardation and early death. And treatment is as simple as a special diet with a low concentration of this enzyme. That's why it's so important if you as a doctor know about this disease, understand what happened with the babies, to put the diagnosis during the first weeks after the born, and only after that you can treat, and these babies will development absolutely normally. Here everything depends from your knowledge. Next amino acid that we will discuss with you, this is a decarboxylic amino acid. Why decarboxylic? Because they have two carboxylic group. We have only two decarboxylic amino acid, aspartate and glutamate, aspartic acid and glutamic acid. Glutamic acid. Glutamic acid is the populous amino acids in our body. Its concentration is very high, especially in the brain, because these amino acids perform various functions. First of all, it plays a very important role in the energy production, since glutamine serves as a supplier of alpha ketoglutarate, a component of citric acid cycle. You know that during the deamination or transamination, glutamate is converted to alpha ketoglutarate. We already discussed today that it's a part of glutathione tripeptide. We start biosynthesis of glutathione from condensation of our glutamic acid with cysteine. And if you remember, we form abnormal bonds between them. This carboxylic group took part in the formation of this bond. Glutamate is glucogenic amino acid, glucose is produced. Glutamate play a very important role in some of our proteins. For example, later we will tell about a special role of it in the activation of blood clotting factors. Under the action of vitamin K, we will attach one more carboxylic group to glutamate, and this gamma carboxyglutamate lead to activation of the formation of this gamma carboxyglutamate, and the blood clotting factors lead to activation of them. Next lecture you will see about a special role of glutamate in detoxification of ammonium. Glutamate is a source of glutamine, which in turn has a special functions in our body, and decarboxylation of glutamate lead to production of GABA. See how many important functions. And let's see this reaction. Gamma amino butyric acid synthesis, GABA synthesis. You see, name of enzyme which took part of this reaction. Catalyze this reaction is glutamate decarboxylase, and usually vitamin B6 is a coenzyme of this enzyme. We produce GABA from glutamate. In this reaction, you see the simplest reaction. Usually you have no problem to write this reaction, and you know that GABA is play a very important role. First of all, this reaction, decarboxylation of glutamate, occurs in the nerve system, and GABA, which we produce, serve as a main inhibitory mediator in the highest part of the brain. The content of GABA in the brain in 10 times higher than other neurotransmitter. I think that you know mechanism of its function. It increases the permeability of postsynaptic membrane for potassium ions, which causes inhibition of the nerve impulse. and increases the respiratory activity of the nerve tissue. GABA improves blood flow to the brain, so it plays very important role for the normal functions of our brain. And deficiency of this enzyme lead to deficiency of gamma aminobutyric acid and many problems in our body. Next decarboxylic amino acid is aspartic acid. No, as usually, it used for protein synthesis. It's a glucogenic amino acid. It is a precursor of asparagine molecules. It the same took part in detoxification of ammonium, and it plays two roles in this functions. First of all, it can take part in unharmful transport of ammonium from the nerve tissue to the liver, and you will see that this is a part of the urea cycle. Aspartic acid, aspartate, took part in biosynthesis of purines and pyrimidines, and I will show you its function in purines I show you before it when we tell about glycine functions, and on the next slide you will see that aspartic acid took part in pyrimidine synthesis the same. It is a donor of the nitrogen in the urea cycle. I already explained it. And of course, you remember that it's a member of malate aspartate shuttle mechanism, which is so important for transportation of NADH2 from the cytosol to the mitochondria matrix. Here you see biosynthesis of pyrimidine rings, and if you see well attention, the most part of this ring, this will be taken from aspartate. So aspartate is necessary and for purines and for pyrimidine synthesis. That's why it's absolutely play a very important role, in biosynthesis of DNA and RNA. We tell till this time about decarboxylic amino acid. Now you can see amino acid with two amino group. See here, 1 second, 1 second. And again, we have only two amino acids, lysine and arginine. Let's tell about a special metabolism and a special function of them in our body. See, arginine, it's a glucogenic amino acid. Of course, we use it for protein synthesis. Arginine, you already know, yes, initiate production of creatine. It is a member of the urea cycle. And you see nitric oxide, the same, it can be produced from arginine. Nitric oxide. Now, I again repeat it. You see, 3 times we see this picture because 3 amino acids took part in these processes. And now we tell about arginine, which is a donor of this amidine group in these processes. Next lecture, you will listen information about the urea cycle and arginine. See here, you will tell about each of these reactions. Especially now I want to show you that arginine, it is a member of this, the same very famous cycle in our body. It's the same Krebs cycle. Moreover, it is the first Krebs cycle. He described it first. And arginine is the part of this cycle. And we already saw that arginine serves as a source of nitric oxide in our body. And nitric oxide, the same is important, you see, source. And nitric oxide is an important signaling molecule that activate formation of the cyclic AMP in our body. And of course, it's very important for the cyclic AMP is the second messenger, and it took part in the formation of the action of different peptide hormones. It the same regulate the heart contractions, regulate the tone of the blood vessels. In addition, the nitric oxide radical is involved in the regulation of the rate of apoptosis, prevent the platelet aggregation and thrombosis such way, regulate the secretion of mediating hormones, and it's very important, it has anti-cancerogenic activity. If we tell about the function of lysine in our body, lysine the same, a member of proteins, and later I will show you that it plays very special and important function in some of our protein. It's a ketogenic, clear, if you remember there are two clear ketogenic, leucine and lysine amino acid, and it is a precursor of carnitine, and I hope that you remember that carnitine took part in transportation of fatty acid across mitochondrial membrane. What is the special role of lysine in our proteins? First of all, in the proteins, we can see not clear glycine, but many derivatives of this amino acid. For example, in collagen, there are very many hydroxylysine molecules. Sometimes we can see methyl lysine or acetyl lysine in the proteins. And lysine can take part in the formation of some lysine itself or its derivatives can take part in formation of additional cross-linked in the collagen, and it's a special role of lysine in the collagen. Detail we will tell about it later when we will study biochemistry of connective tissue, but already now I can to show you that formation of these additional bonds, additional cross links lead to increase of and it's very important, it's even essential for the tensile strength of the collagen. If we tell about another protein of connective tissue, elastin, lysine and here plays a very important role because lysine took part in the formation of desmosine. It's a special structure. You see, this is a desmosine. Desmosine is composed of 4 lysine residues and allowing for bonding to the multiple peptide chain. The 4 lysine groups, you see, lysine groups combine to form like pyrimidine nucleus, and desmosine is found uniquely in elastin, and these elastic properties of elastin it's a pair only due to desmosine. Desmosine gives this elasticity. And you know that elastin, it's a part of many our connective tissue and in the skin and lungs, and very important for the elastic arteries. That's why the desmosine structure is rather important for the normal functions of these organs. Lysine, the same is involved in the formation of insoluble fibrin. You see that this is fibrin 1, fibrin 2, and the strands of this fibrin polymer are different with other amide covalent bonds between the lysine residues of one fibrin polymer molecule and the glutamine residues of other. And due to condensation of to this molecule, we you see additional cross-linked between these fiber strands, and it leads to formation of the strong three-dimensional network, which includes and platelets and erythrocytes and leukocytes. That's why you see that lysine the same play a very important role in the blood coagulation. And now, finally, we will tell about the metabolism of phenylalanine and tyrosine. Phenylalanine is essential amino acid because its benzene ring cannot be synthesized in our body. Tyrosine is conditionally non-essential amino acid because a part of tyrosine can be produced from phenylalanine. The content of these amino acids in the food proteins and in animals food and in plant proteins, including plant proteins, is quite high. If we tell about phenylalanine, phenylalanine is consumed in two ways. It includes in our proteins. So phenylalanine is essential. We can take it only from the food proteins, and it is can use only or for biosynthesis of our own proteins or for production of tyrosine. And that's all. Phenylalanine has only these two functions. But tyrosine we can take and from our food and from phenylalanine. Tyrosine has many different functions in our body. Its metabolism is more complex than phenylalanine. Of course, tyrosine we can use for not can, but always we use for biosynthesis of our own protein. But together with this, tyrosine is involved in the synthesis of variety biological important compounds like catecholamines, such as epinephrine, norepinephrine, dopamine, thyroid hormones T3, T4, and pigment melanin. In the liver, during the course of degradation, tyrosine and through tyrosine phenylalanine the same, they are converted to metabolites which can serve as a precursor of glucose, because during catabolism we can cleave them into part, the carbon skeleton. One part we can use for glucose synthesis, another part is it converted to acetoacetate ketone bodies. That's why both of them they are mixed glucoketogenic amino acids. Or they can be catabolized to a final product, carbon dioxide and water with energy production. You see, this pathway is rather long, and there are several hereditary diseases associated with the defect in the enzymes of phenylalanine and tyrosine metabolism in different tissue. And we will tell about some of these diseases. First of all, let's tell about the first reaction, formation of tyrosine. In the conversion of phenylalanine to tyrosine, the reaction involves the incorporation of one atom of oxygen into para position of phenylalanine, and the second atom of oxygen is converted to the water. An enzyme which catalyzes this reaction is phenylalanine hydroxylase. Please remember the name of this enzyme. After that, tyrosine already, you see, can be converted to different substances. We already tell about them. Thyroid hormones, catecholamines, or full catabolism of it to or carbon dioxide and water, or we can produce glucose or ketone bodies from them. Now let's tell about the role of this tyrosine in the synthesis of different these important substances, and we start from the biosynthesis of thyroid hormones. In the thyroid gland, these thyroid hormones are produced: thyroxine or tetraiodthyronine and triiodthyronine T3 and T4. They are synthesized from tyrosine residues in the long protein thyroglobulin. This protein is rich of tyrosine residues. First of all, happens the iodinization of this tyrosine in these thyroglobulins, and a special enzyme, thyroid peroxidase, catalyze this iodinization. Iodinization can occur in different manner. We can attach only one iodine molecule or two iodines atom or two iodines atoms, like on this picture. After that happens the condensation of two iodinization tyrosine, and here you see the same can be combination. If we combine two tyrosines with two iodines, here and here, we produce thyroxine molecule. If here, for example, if here attach only one iodine, but here two, and we combine them, in this case we produce triiodthyronine molecule. After that happens the releasing of these hormones from thyroglobulins, and all of these reactions occurs under the action of thyroid peroxidase enzyme. Now, biosynthesis of catecholamines. By convention of tyrosine into catecholamines occurs in the adrenal gland and nervous tissue. In these organs, tyrosine, you see, has not very long but very interesting and important pathway. Why name is catecholamines? Because the name catechol refers to the dehydroxylated phenyl ring, and they have amine, you see, derivatives. That's why full name is catechol amines. Synthesis of catecholamines involves in the, you see, these four reactions, and we produce all of these substances. They're important: and dopa, dioxyphenylalanine, and dopamine, and norepinephrine, and epinephrine. Now let's tell about each of these steps especially. First of all, tyrosine is hydroxylated to 3,4-dehydroxyphenylalanine, deoxyphenylalanine, deoxy, because we have two oxy group, two hydroxy group. That's why DOPA. This is phenylalanine with two oxy group. The name of enzyme which catalyzes this reaction is tyrosine hydroxylase. Second step, during the second step, this DOPA is converted to dopamine due to its decarboxylation, and name of enzyme is DOPA decarboxylase. Like in all other amino acid decarboxylases, coenzyme of this enzyme is pyridoxal phosphate, vitamin B6. See here. In the nerve system, dopamine plays a special role. It's a neurotransmitter, and maybe it's already the final step of reaction. And pay attention on this enzyme, because deficiency of this enzyme can cause Parkinson's disease. This reaction is irreversible. Next reaction. Pay attention on this enzyme. Remember it, please. Deficiency of this enzyme lead to Parkinson's disease. Next reaction, which happened with dopamine, is hydroxylation. And we pair one more third already OH group near this carbon. And the name of enzyme is dopamine beta hydroxylase. For this reaction is necessary vitamin C. See how many vitamins necessary. And of course oxygen. And we produce norepinephrine or noradrenaline. In this reaction, the same is irreversible. And at last, the last reaction in this pathway is methylation of this nitrogen. See, a pair, compare methyl group, and we produce already epinephrine. The differences between epinephrine and norepinephrine is only this methyl group. And what is the donor of methyl group? Of course, our methionine, active methionine. So one more amino acid took part in epinephrine synthesis, and the name of this amino acid is methionine. SAM is a donor of this methyl group. That's all. You see that these reactions, they are absolutely not harmful, and you already know the mechanism of each of these reactions. Of course, catecholamines plays many important. They play many important functions in our body. They regulate our metabolism and carbohydrates and lipid metabolism, and we tell about these functions of them many times. They stimulate the degradation of triacylglycerol and glycogen, first of all. Now, but together with this, they increase the blood pressure. Dopamine and norepinephrine serves as a neurotransmitter in the brain and autonomous nervous system. And as we already tell, that deficiency of dopamine, problem with dopamine synthesis, can cause to Parkinson's disease. Parkinson's disease is one of the most common neurological diseases. Frequency is very high, 1 to 200 among the people over 60. And the disease is accompanied by three main symptoms: ataxia, stiffness of movement, rigidity, muscle tension, tremor involving involuntary movement, mask-like face, and lethargy. Treatment is a problem because dopamine does not cross the blood-brain barriers, and that's why it can't be used as a drug. We can use DOPA, and really we have levodopa, for example, medicine, and it's useful in the treatment of Parkinson's disease. And really, in the brain, DOPA very fast is transformed into dopamine due to decarboxylation. And it lead to elevates symptoms of this disorder. But unfortunately, we have a negative effect because dopamine synthesis occurs not only in the nerve tissue, but in other tissue the same, and it leads to many negative effects. It's such as nausea, vomiting, hypertension, and other. Sorry. Now, one more product can be produced from tyrosine in our body. In the pigment cells, melanocytes, tyrosine acts as a precursor of dark pigment melanin, and you know that melanin it is responsible for the color of our skin, hair, eyes, retina. Biosynthesis of melanin is rather complex. Only pay attention that during the first reaction, again we produce DOPA, like in epinephrine synthesis. Only we use another enzyme. If in epinephrine synthesis we use enzyme tyrosine hydroxylase, in melanin synthesis, name of enzyme is tyrosinase. Not need to study for these long and complex multi-stage branched processes. Only remember the name of this first enzyme in this long pathway, tyrosinase. And if we tell about the deficiency of this enzyme, it leads to deficiency of melanin synthesis. And name of this pathology processes is albinism. Um, Albinism, what is a clinical manifestation? Even name already explain everything, because from Latin albus means white. And really this is the absence of pigmentation of skin and hair and eyes, and patient often have a reduced visual acuity, photophobia of course appears, and for them the sunshine is very dangerous. The prolonged exposure of this patient to the open sun leads to skin cancer. The incidence of the disease is approximately 1 to 20,000. You can see these two boys with albinism, but and so here only problem with the pigmentation, and of course try to avoid the action of the sunlight. Absolutely okay with their mentality, and they have no any other pathological problems in their life. Only this problem with pigmentation. But if we tell about deficiency of this first enzyme in phenylalanine metabolism, phenylalanine hydroxylase, I ask you to pay attention on this enzyme. Here, in this case, a pair disease with many big problem already. The name of this disease is phenylketonuria, famous PKO. It's the same as inherited disease, and phenylketonuria is very dangerous for without treatment. It can lead to full mental retardation. You see that we already tell that phenylalanine has only two ways in our body, or protein synthesis or tyrosine synthesis. And deficiency of this enzyme, phenylalanine hydroxylase, lead to what? Deficiency of tyrosine, and as a result lead to problem with production of all these substances from tyrosine and accumulation of phenylalanine in our body. Concentration of phenylalanine becomes high. Phenylalanine is hydrophobic. That's why our body try to look for any another way of increasing of its solubility for the excretion from the body. And we use transamination reaction. Due to it, we change this methyl group on keto group and produce phenyl pyruvate. Really, in the liver, even a healthy people, very small amount of phenylalanine, less than 10%, can be converted to phenyl pyruvate. Only concentration of them is very low. But during this pathology condition, this way becomes main way of the metabolism of phenylalanine, and it lead to increasing of phenyl pyruvate, sometimes in 2000 times. And in the blood appear so many rather dangerous substances, because after that phenyl pyruvate can be converted or into phenyl lactate due to the reduction reactions, or due to oxidative decarboxylation into phenyl acetate. And increasing of concentration of all of them in the blood and a pair of them in the urine, it's symptoms of this disease, and even they give the name of this disease, phenylketonuria. urea. And the most severe manifestations of the phenylketonuria are impaired mental and physical development, convulsion syndrome, pigmentation, of course, disorder, because with the low tyrosine happen the problem with melanin synthesis. And if untreated, patient do not live up to 30 years. And incidence of the disease is rather high. It's 1 to 10,000 of the newborn. The disease, we already tell, it's inherited, and it's inherited in autosomal recessive manner. Severe of PKU manifestation is associated, first of all, with the toxic effect on the brain cells, a high concentration of phenylalanine, phenylpyruvate, and phenylactate. High concentration of phenylalanine limits the transport of tyrosine and tryptophan across the blood-brain barrier and inhibits the synthesis of main neurotransmitters, dopamine, norepinephrine, serotonin. Progressive impairment of this mental retardation and physical development in the children with PKU can be prevented by the diet with the dosage of phenylalanine. Only if this treatment is started immediately after baby is born. And brain damage can be prevented in this case. Only in this case is important the earliest diagnosis of phenylketonuria. To diagnose of phenylketonuria, we can use different methods, and qualitative and quantitative methods of determinant of pathological metabolites and in urine and in the blood. The populace now is using of the Guthrie test. The Guthrie test is a semi-quantitative test. And what happened how we do this test? You see that a drop of blood is obtained usually by pricking the heel of a newborn infant in the hospital, of course, but we do it not immediately after birth, burn, but on the second or third day of the life, necessary to give some time for the accumulation of phenylalanine in this body. The blood after that is collected on a piece, you see, of filter paper, and after that mailed to any laboratory. This small disc, you see, of sample with the filter paper, after that punched out and placed on agar gel phenyl plate, agar gel plate, you see, with Bacillus subtilis. And these bacterials, for these bacterials, phenylalanine, the same, is essential amino acid. They have to take them from environment. That's why around the disc with extra phenylalanine, the bacteria grow more intensively. You see, and this is a positive result. So babies, these babies with this blood has a phenylketonuria, very high concentration of phenylalanine in the blood. This test is high specific. Possible to use another test, possible to determine the phenylpyruvate in the urine with the using of ferrichloride test, and green color is obtained. We will do this test during our classes. You will see this test. By this test is not specific, and since many other compounds gives false positive tests. After that, if we understand that this baby has this problem, necessary immediately start to treat this baby with a special diet with the lack of phenylalanine. It's absolutely synthetic food. You see that all babies with phenylalanine without treatment, they have mental retardation, albinism, and one of symptoms, it's a special mouth smell of urine. High concentration of these phenylketone bodies in the urine give this special smell. And you see, necessary immediately start to treat this baby and we can do it with this special diet with a low concentration of phenylalanine. If we start to treatment immediately, brain damage can be prevented. And now it is believed that dietary restriction can be eased after 10 years of age. The brain myelination is end. But nowadays, many pediatricians are learning that towards the lifelong diet, and I think it is correctly better to prevent accumulation of phenylalanine in the body. And you see, pay attention that concentration of phenylalanine must be reduced, but not absolutely eliminated, because these essential amino acids have to be used for protein synthesis. First of all, and of course, this food must be rich of tyrosine. Tyrosine must be enough for biosynthesis of all necessary substances, hormones and melanin and so on. In this case, this baby can develop absolutely normally. The defective gene responsible for phenylketonuria can be detected in phenotypically normal heterozygotes carriers using the phenylalanine tolerance test. For this, the subject is given on an empty stomach about 10 grams of phenylalanine in the form of a solution. And then every time blood samples are taken, and we check concentration of tyrosine in these samples. And tyrosine content, normally concentration of tyrosine in the blood after phenylalanine loading is significantly higher than in heterozygotes carriers of this phenylketonuria gene. Because these heterozygotes carriers cannot normally produce tyrosine from phenylalanine. And this test is used in genetic counseling to determine the risk of having a sick child. If we tell about catabolic pathway of phenylalanine and tyrosine, you see that phenylalanine is converted to tyrosine. After transamination of tyrosine, we produce hydroxyphenyl pyruvate. After some reaction, this hydroxyphenyl pyruvate can be converted to homohentisic acid, which in turn is converted to due to oxidation to 4-malonyl acetoacetate. Under the action of, pay attention on this enzyme, homohentizate dioxygenase or homohentizate oxidase. You see this enzyme. Pay attention on this enzyme because deficiency lead to one more problem, and we will tell about this problem a few seconds later. Now, isomerization of malonyl acetoacetate lead to production of fumaryl acetoacetate. Pay attention with different color. I show you the position of each group in each of these molecule. And now, when we break down this molecule in two half compounds. From this part we produce fumarate, from this part acetoacetate. Fumarate can be precursor of glucose. Acetoacetate is ketone bodies. That's why they are glucoketogenic. Now, here may be, of course, deficiency of different enzyme, but we will tell about deficiency of homogentisate dioxygenase. Deficiency of this enzyme lead to alkaptonuria. If we can see block here, it lead to what? Accumulation of homogentisate in the body. And under the action of air oxygen, this homogentisate can be converted of black alkapton, which can be excreted with the urine and gives to urine black color, dark urine. You see, from here, alkapton, urea. In the urine appear alkaptons. Is it dangerous? Not so high like phenylketonuria. This disease is characterized by except formation of the dark pigment urine, we can see accumulation of the pigments in the some part of the body. You see, it's a cosmetologic effect, but the same it leads to pigmentation, you see, of connective tissue. You see, name of this is ochronosis, and can lead to arthritis. The frequency of this disease is not very high, about 5 causes per 1 million of the newborns. This disease is inherited and in the autosomal recessive manner, and treatment is usually no treatment of this disease. Maybe diet with low proteins, especially phenylalanine and tyrosine, and that's all. And that's all. You see here, usually only arthritis and all these cosmetology effect, and that's all. This is alkaptonuria. We discussed with you all materials for today. Thank you for your attention. Goodbye, dear students.